We're going to talk about G-protein-linked receptors in this video. When a G-protein-linked receptor is bound by its hydrophilic messenger, the G-protein is activated and it can go and do one of two things. So it can go bind to an ion channel and open or close the gate, or it can go bind to an enzyme and turn the enzyme on, which can produce second messengers. So if the G-protein goes and binds to an ion channel, the ion channel is called a slow ligand-gated ion channel, and that's because it's slow to open and close. So let's just take a look at a picture of this. So the hydrophilic messenger will bind to this G-protein-linked receptor. That'll activate the G-protein. Part of the G-protein will take off and then go and bind to this slow ligand-gated ion channel. And we'll open the gate, and that'll let the specific ion in or out of this slow ligand-gated ion channel. So the slow ligand-gated ion channel is slow to open because it takes some time to activate the G-protein once the hydrophilic messenger binds to the G-protein-linked receptor. And then also this slow ligand-gated ion channel is going to stay open longer because the G-protein is going to stay attached for a little while, and then once it falls off, then the gate will close and then the ions will stop moving in or out of the cell. And then those ions moving in or out of the cell will cause the cell response. The other thing that the activated G-protein can do is bind to an enzyme and then turn that enzyme on, and the enzyme will produce second messengers. The second messengers that could be produced by the enzyme, there's actually 5 different ones, so make sure you know these. So these are important to know. So we've got cyclic AMP, cyclic GMP, DAG, IP3, and calcium ions. And I'm just going to use the abbreviations on the exam. I won't make you spell these out. So this is table 5.3 from your book. I would be familiar with this. So it lists all 5 of the second messengers. And we'll go through and talk about where these second messengers come from and then what they do. And you don't need to know examples of these. You don't need to know this last column. All right, let's take a look at our first second messenger pathway. This is the cyclic AMP pathway. So we have our hydrophilic ligand bind to our G-protein-linked receptor there. And that's going to activate the G-protein. So what happens is we get GTP that ends up replacing the GDP that's there. And then part of the G-protein will take off. And then it's going to go bind to this enzyme in the cell membrane called adenylate cyclase. So there's an L there. And then that'll turn on the adenylate cyclase enzyme. And adenylate cyclase will grab some ATP, and it's going to turn the ATP into cyclic AMP. And cyclic AMP is a second messenger. And then cyclic AMP will then activate protein kinase A, which is an enzyme. And the A just matches the A in adenylate cyclase. And remember that kinase enzymes phosphorylate things. So being a protein kinase, it's going to phosphorylate proteins. So it's just going to grab a protein and on some ATP. That's where it's going to get the phosphate group from. And then it's just going to stick a phosphate group onto this protein. And then these phosphorylated proteins will cause the cell response. Okay, and then we need to be able to turn the pathway off. So how we do that, there's a couple of ways. So one is the hydrophilic ligand. That's actually our first messenger. That will fall off the G protein linked receptor, because remember it binds reversibly to these protein receptors. So when it falls off, it'll stop activating the G proteins. And then the cyclic AMP, our second messenger, we can deactivate that with an enzyme called phosphodiesterase. And phosphodiesterase is just going to break the circle that's in cyclic AMP and just make it into plain old AMP. And then these phosphorylated proteins that are causing the cell response, we can dephosphorylate them. So we can remove the phosphate group. And remember, the enzyme that dephosphorylate things is called phosphatase. Okay, so questions that I can ask you on the exam. I won't make you draw this out, but I could ask you questions like, out of this pathway, what's the second messenger? And you'd have to pick out, oh, that's cyclic AMP. Or I could ask you, well, what does cyclic AMP do? Well, it activates protein kinase A. So those are some of the kind of questions I could ask you about this. And if you practice drawing this, and if you could draw all this out and not look at your notes and be able to do that, then you could get any question I would ask on this correct. All right, so I would practice drawing this out, and then anything I ask on this, you would be able to get right. And then this is just a nicer picture than what I drew just from your book. And again, just showing that you've got your G protein linked receptor here. The hydrophilic messenger binds to it. We activate our G protein. Part of it takes off. We'll turn on our enzyme adenylate cyclase. And that adenylate cyclase will take the ATP, turn it into cyclic AMP, which is our second messenger. And then cyclic AMP will turn on the protein kinase A. And then protein kinase A will phosphorylate proteins, and then the phosphorylated proteins will cause the cell response. And then turning this off, our first messenger can fall off the receptor. That'll stop activating the G proteins. We can get rid of the cyclic AMP with this enzyme phosphodiesterase, which chops the circle, and you just get the cyclic AMP. And then we can dephosphorylate the proteins with that enzyme phosphatase. Okay, and then just giving you an example of the cyclic AMP pathway. This is the hormone ADH that's produced by the posterior pituitary in the brain. And ADH stands for antidiuretic hormone. So this hormone is going to go to your kidneys and tell you to save water from the urine and put it back in the blood. Okay, so then that'll make you urinate a smaller volume of urine. So you would want to secrete this hormone like maybe you're dehydrated. So you want to save some of that water and don't pee that out. Okay, so we've got ADH, which is a protein hormone. It's hydrophilic. Here's our G protein linked receptor. cell surface receptor here, ADH binds. We activate the G protein. It's going to turn on that enzyme adenylate cyclase, which will take the ATP, make the cyclic AMP our second messenger, and that'll turn on the protein kinase A enzyme. That'll phosphorylate some proteins, which will cause the cell response, which is to put some aquaporins or some water channels out into the apical membrane of the epithelial cell that's lining the renal tubules in the kidneys, and that will allow water to be reabsorbed and put from the urine back into the blood. So that's how ADH works. Okay, another pathway using these G protein-linked receptors is the cyclic GMP pathway. And if you've already memorized the cyclic AMP pathway, this one's easy. Anywhere you see an A, you just replace it with a G. So this one, we're going to use guanylate cyclase as our enzyme. And when it gets turned on by the activated G protein, it'll take GTP and make our second messenger cyclic GMP. And then cyclic GMP will activate the enzyme protein kinase G. And then protein kinase G will phosphorylate the proteins, and that'll cause the cell response. Okay, and then our third pathway that involves these G protein-linked receptors is this phosphatidylinositol pathway. I won't ask you that name because that's pretty horrible. So with this one, we've got our hydrophilic ligand binding to our G protein-linked receptor. And remember, this is our first messenger. And that will end up activating our G protein. It'll take off and it will activate an enzyme in the cell membrane called phospholipase C. And this phospholipase C enzyme is going to grab one of the phospholipids in the membrane called PIP2, and it's going to break it apart into two pieces. So we're going to break it into DAG, which is a second messenger. And then we're also going to break it into IP3, which is another second messenger. Okay, and DAG and IP3 are going to do different things. So DAG will activate protein kinase C. And the C just matches the C and phospholipase C. And being a kinase enzyme, remember, it's going to phosphorylate proteins. So we're just going to take a protein and some ATP, and we're going to stick a phosphate group onto the protein. And then those phosphorylated proteins will cause the cell response. So that's just like we've been talking about. Okay, but the IP3, the other second messenger, will do something really different. So IP3 will go over and bind to a calcium ion channel in the smooth ER, open the gate, and in the smooth ER we have stored some calcium ions. And when you open that calcium channel, calcium will come out. And the calcium is our third second messenger in this pathway. So we've actually got 3 second messengers in this pathway. So we've got our DAG, IP3, and then calcium ions. The calcium ions by themselves could cause a cell response, depending on the cell, or they can go bind to a protein called calmodulin. And that'll activate the protein. Oops, spelled it wrong. And then that'll activate that calmodulin protein. And then that will activate a protein kinase, which will then phosphorylate proteins. And then those phosphorylated proteins will cause the cell response. Okay, so this one's got a lot more going on. But again, if you draw this out a couple times, you'll have this down, and any question I ask on this, you could answer. And again, questions I could ask. I might ask, what does the enzyme phospholipase C do, for example? Okay, so remember phospholipase C will take that membrane phospholipid PIP2 and split it into DAG and IP3, which are both second messengers. Okay, or I could ask, what are the 3 second messengers produced in this pathway? So DAG, IP3, and calcium. Okay. So those are just some examples of questions I could ask here, right? And this is a nicer picture than what I drew from your book. So it just shows the hydrophilic messenger binding to the G protein linked receptor, activating the G protein. Part of it takes off, turns on the phospholipase C enzyme, which will grab the membrane phospholipid PIP2, split it into the 2 second messengers, DAG and IP3. DAG will activate protein kinase C, turn that enzyme on. Protein kinase C will phosphorylate proteins, and that'll cause the cell response. IP3 will go over and open calcium channels in the smooth ER. We'll let calcium ions out into the cytoplasm. Calcium ions by themselves can cause a cell response, or they can bind to the protein called calmodulin, which will activate it. And then the calmodulin can turn on the protein kinase enzyme, which will then phosphorylate proteins, and then that can cause a cell response. All right, so I know these G protein linked receptor pathways are pretty complicated, but why do cells like them? So notice that we've got all these steps, and you get this cascade, and with each step you get more things produced, and you end up amplifying that first initial signal. So let's take a look at some pictures. All right, so from our first hydrophilic messenger here, we've just got one of those. You end up from one hydrophilic messenger, by the end, you end up with 2.5 million phosphorylated proteins to cause a really big cell response. Okay, so you can really amplify that first messenger, and you can get a huge cell response from that. Okay, so that's one of the advantages of using these second messenger systems with the G protein linked receptors. Okay, and here's just another picture of this, just showing you this cascade, and each step you end up getting more things generated, and you end up from one signal molecule up here, you end up really amplifying that signal, and you end up with all these phosphorylated proteins down here, and that'll generate a really big cell response in the target cell.